Hi there, Year 12. This video we're going to give you your feedback for the multiple choice section of your trial HSC for biology. Now, hopefully by now you've received your paper with your answers and marks on it. And so this will go through the multiple choice questions, explain to you why the answers are the answers that we're looking for, and give you the correct answers if you're not sure. Now, just some general multiple choice feedback first of all. This was an area where a lot of the groups separated themselves in terms of the final marks, and it's really common that multiple choice does this in the HSC. In fact, the majority of band 6 or the high mark students tend to lose most of their marks in the multiple choice section. So if we look at just the top 15 students in terms of the raw marks for this particular trial, there was a 9-mark spread between the student who got the top multiple choice mark, which I believe was about 19, and the student who got the lowest multiple choice mark, which was 10. And that's for the top 15 students. So a couple of extra multiple choice marks either way might have made a big difference to the overall mark in the end, and certainly could have made a difference in the overall rankings as well. The next 15 students after that, taking us sort of to the middle group of the trial results, there was a 7-mark spread in the multiple choice questions. The average mark for the multiple choice was 13 out of 20, and it was actually the 17th best section of the paper overall, meaning there was 16 other sections or individual parts of questions that were answered better than the multiple choice. So again, this is an area that we can focus on leading into the HSC itself, and hopefully by giving the feedback here on how to read these questions and how to answer them, will give you some guidance when it comes to the HSC later in the year in how to tackle these multiple choice type questions. So let's start with question 1. The diagram below is a female human reproductive system. Where does fertilization occur? So we've got the options here, A, B, C, or D. You should know that human reproductive system and fertilization is what the question's asking about. You should know that in fertilization, the sperm enters through here and the egg is released through there. The sperm is going to travel down the fallopian tubes here at the same time as the egg is going to travel down the fallopian tube. And so fertilization is actually going to occur in the fallopian tube before that zygote then moves down and gets implanted in the uterus. So the answer we're looking for here was answer A. All right, question 2. Again, in humans, innate immunity includes. So innate immunity is the immunity that responds the same to no matter what the pathogen happens to be. It's not trained, it's not acquired, it's not anything that your body has learned to deal with. It's just the basic second line immunity that we have once that pathogen has breached our protective barriers. So if we look at these options here, anything that has B or T cells is not going to be part of innate immunity. So we know that this production of memory B and T cells and activation of B cells by helper T cells, B and C are not going to be an option that we can choose for this one. Antibodies are the job of the memory B cells and the B cells, the plasma B cells rather, and that's part of the third line or the specific immunity or the specific response. So again, A can't be the option, which leaves us D, and macrophages detecting pathogens and removing them by phagocytosis. That is very much an innate immune response. Macrophages are swooping around all the time in our body and picking up any pathogens or anything that detects as foreign and eating it regardless of what it is. So it's not specific, it's not acquired, that's purely innate. All right, question 3. Below is a description of 3 pathogen types. Now, pathogen A, we needed to break apart a little bit of what these things were to give us some clues as to what they would be. So pathogen A reproduces asexually. It's between 2 and 1,000 microns in size, and it's a eukaryotic cell. So eukaryote cells are cells that have a nucleus. So pathogen A, B, and C respectively, we're looking for the A ones here that might have a nucleus and that might reproduce asexually. Now, protozoa, they're eukaryotic, they have a nucleus, they reproduce asexually, so that could be an option A. Macroscopic parasite, they don't reproduce asexually generally, or not as a standard. So B and C, we can rule those ones out straight away. So now we're choosing between A and D. Now let's look at option B here, or pathogen B. So these are all the key points of that. It's prokaryotic, it's binary fission, it doesn't have a nucleus. That fits bacteria. So we still have the option of A or D, protozoa and bacteria, in both of these. So it's going to come down to the prion or the virus. We need to determine whether C is a prion or a virus. Now again, key points here, very small. It's acellular, containing a protein coat. Now prions are protein diseases, or diseases of proteins, but they're not acellular. The protein coat is actually a protective thing that the virus has that protects it when it's outside of the host. And this is the other key one here, reproduce inside a host cell. That is very typical of a virus, which means our answer is going to be D. All right, question 4. The diagram below is the reproductive structures of a flower. If a botanist wished to cross-pollinate a flower, how would they ensure that self-pollination did not occur? So in other words, how do we make sure that the pollen that this flower produces doesn't self-pollinate itself? So the pollen is going to land here on the stigma. And so how do we make sure the pollen of this plant doesn't land on that stigma? We need to remove the parts that produce the pollen for that flower. And in this case, that's the anther. So option A, removing these here, the anthers, is going to be the correct answer. If we remove those anthers, there's no pollen left in the flower. It's not going to accidentally self-pollinate when we're trying to cross-pollinate it with something else. All right, question 5. Which of the following is an example of an allele? An allele is a trait variation. And so let's look through all of these different options here. So short-sightedness and wearing contact lenses. So contact lenses can be a treatment for short-sightedness. You don't have a variation in anyone that's short-sighted or contact lenses. That's not even a genetic thing. It's a treatment. So we can rule out A as being a possible choice. White fur in mice and curly hair in guinea pigs. Now, you could make an argument that white fur and curly hair are potentially alleles. In fact, they would be alleles. But here we're looking at mice versus guinea pigs. So when we look at alleles, we look within the one species. So we can say that B is not the answer. Brown hair and blonde hair colour doesn't tell us what species we're talking about here, but we have colour variations of the same thing, so colour variations of hair. So different traits of colour for the hair, that definitely fits the idea of the allele. But let's look at D just to make sure. Codominant feather colour and sex-linked haemophilia. So codominant is a pattern of inheritance of alleles, sex-linked is a pattern of inheritance of an allele, but they're not examples of alleles specifically. So that means the answer here we're looking for is answer C. Brown hair and blonde hair are different alleles. The alleles here relating to the different traits that are produced in the hair colour in this case. All right, question 6. DNA profiling uses polymerase chain reaction and gel electrophoresis, and this allows the comparison of which of the following. So the key things here are PCR and gel electrophoresis. Now in class we would have done a prac of gel electrophoresis where we separated fragments to see how they spread through the gel. And when we think of gel electrophoresis, we're not really identifying genes. We're not really screening or determining entire genomes. We're not able to detect specific sites of mutations, but what it does do, gel electrophoresis and PCR as well, is it splits the DNA up into fragments based on their size, and those fragments then can be analysed to determine genes and mutations and those sort of things as well. So the answer we're looking for here is C. All right, question 7. Antibiotics are ineffective against viruses because viruses do which of the following? So antibiotics, you should know, work sometimes well on bacteria, but they don't work for viruses at all. And to answer this question, it would have helped if you could understand how it is that antibiotics impact bacteria, but you could have probably worked it out by first principles anyway. So let's look at option A. Keep changing their external protein coat. So viruses keep changing the external protein coat. That's why antibiotics don't work on them. That is not an option. The external protein coat is a protective part of the virus, but changing their coat is not going to have any impact on the way the antibiotics work. Viruses are not living cells and thus do not metabolize. Well, part of the way antibiotics work is by causing metabolism of the chemical, which then kills off the bacteria or those type of cells. So that is the option there. That is the correct answer. But let's look at option C. Can change antibiotics into useful food substances. Viruses, they're not living, they're not really eating things. They're just packages of DNA or RNA that are highly skilled at reproducing themselves. So they're not really eating, so we know C is not an option. And are able to disguise themselves in the host cell membrane. So again, that is something that viruses do, but that's not related to antibiotics. Again, that's a mechanism to hide from the immune system, not to hide from antibiotics. We've already identified that B is the correct answer, but hopefully if you weren't quite sure or couldn't quite remember how antibiotics work, you could have worked that out through first principles. All right, question 8. Which of the following represents the sequence of events in the production of a protein from the genetic code? Now, this question, you needed to understand the genetic code is the DNA, and we're looking for the production of a protein from that genetic code. So it has to be one that starts with DNA. It's going to be A or B. Now, the DNA, the first process of that is the transcription phase, which involves the mRNA. So that means that B already is the only possible option we have. And so if we look at a production of a protein, we go from DNA into messenger RNA. That then codes for the amino acids. The amino acids then join together to form the protein. So B is the answer that we're looking for there. All right, question 9. The flowchart represents some of the stages of an application of a beneficial biotechnology. And so here we're looking at acquiring a gene or targeting a gene, extracting that gene, and we've got a plasmid from a bacterial cell as well. And stage X is where we're going to join these together. And then the final part is insert plasmid into host bacterial cell to produce a GM organism. So this is the process that we might use for producing insulin using bacterial cells or bacterial plasmids. So a suitable description for stage X would be, let's have a look. Insert bacterial plasmid into the required gene. Well, that's the opposite of what we're trying to do. We want to put the gene into the plasmid, so A can't be an option. Insert bacterial plasmid into source chromosome. So again, we're not inserting the plasmid into the genome. We're inserting part of the gene into the plasmid. So B can't be an option. Insert required gene into host bacterial cell. Well, that's going to be the end idea of what we want to do, but we're going to use this plasmid to do it, which means D has to be our answer. Insert required gene into bacterial plasmid. Question 10. The following flow diagram relates to blood glucose regulation. So blood glucose regulation is all about the pancreas and the liver, glucagon and insulin, and how that all works together. So we have organ X that releases hormone Y that acts on organ Z and promotes conversion of glycogen to glucose. So organ X and organ Z, one of them is probably going to be the liver, one of them is probably going to be the pancreas, and releases hormone Y. We need to remember which hormone it is that changes glycogen into glucose. Now, hopefully you remember that when we have too much glucose in our blood, then we release insulin to turn that sugar or that glucose into glycogen. So insulin is one of the options, but we're looking at the reverse of this here. So anyone that has insulin as the option is going to be incorrect in this case. And the question is all about the blood glucose regulation. I've mentioned organ X before, and releasing the hormone Y. So these organ X and hormone Y are what we're looking for. Now, I've already identified that insulin is not going to be the one that we use to convert glycogen into glucose. What we're looking for there is the other one. So our options are going to be glucagon. So glucagon has to be either B or D as the options, because that's the only one that hormone Y can be to change glycogen into glucose. Now, then we need to determine whether organ X is going to be the liver or the pancreas. Now, you might be easily mistaken into thinking that the liver is organ X, so that's where the hormone comes from. But actually, it's the pancreas that releases glucagon, that then so the pancreas will release glucagon, which acts on the liver to promote that conversion of glycogen into glucose. So the answer we're looking for there is D. All right, question 11. The figure below shows some changes that occur during the menstrual cycle. So we're looking at the menstrual cycle here, or the ovulation cycle, and which hormones cause thickening of the lining of the uterus. Now, this is a question where the graph here may have helped you if you weren't quite sure. And to answer it, you need to understand when it is that the uterus lining is being thickened and why that is happening. Now, it's happening so that by the time we have ovulation, we have a thick enough lining of the uterus that hopefully fertilized egg or zygote can implant and then be carried through for the pregnancy term. So we're looking for one of these here that's going to peak probably before ovulation. Now, LH is luteinizing hormone, FSH is follicle stimulating hormone. Both of those are related to the ovulation phase itself, whereas the estradiol, or the estrogen, is the one that's going to be related to the thickening of the lining of the uterus. So the answer for that one then is estrogen. All right, question 12. An epidemiological study is concerned with which of the following? So epidemiological study is the key thing here that we're looking for. And epidemiological studies are all about working out frequencies and, how common the disease is, where that disease is, what are the risk factors and what are the causes of the disease. So if we look at option A, the cause of diseases and how to cure or control them, that could be part of an epidemiological study, or an epidemiological study might help identify those, but it's not the most complete answer option that we have here. Finding the causative pathogen of a disease. Now, again, epidemiological study is probably not what we're looking for there. We're probably more looking for a clinical trial of some sort, something more of a direct comparison, although an epidemiological study might clue us in that there's some sort of pathogen that's causing the disease. Option C, the frequency and geographic distribution, causes and risk factors of a disease, that very much is in the epidemiological study realm. So that's the correct answer we're looking for there. Collecting the data about the prevalence of a disease in a community. Again, that would be part of an epidemiological study potentially, but it's not the entire picture of an epidemiological study. So the best answer here is answer C. All right, question 13. Which example below shows the development of a transgenic organism in agricultural or medical applications which increases biodiversity? So transgenic organisms, what we're looking for. And in this case, we're looking for one that's going to increase biodiversity. Now, increasing biodiversity is going to be the key thing here in terms of determining which answer is correct. So cloning embryos, which can be used as a source of stem cells to make new heart muscle. Anytime we're cloning, we're making an exact copy of something we already have. So that's not going to increase biodiversity at all. So we know that A is not an option in terms of the answers. Inserting a gene from maize and a gene from bacteria into rice to produce golden rice. So here we have a transgenic organism, rice, where we've got a gene from bacteria and a gene from maize. That definitely is going to increase biodiversity because we're increasing the genome, or the potential genotypes of this particular rice, by including more possible genes in that combination. So B is the answer we're looking for, but let's just look at C and D as well. Somatic cell nuclear transfer to produce a cow with greater milk production. Somatic cell nuclear transfer is just cloning, like with Dolly the sheep. So that, again, complete copy of the cow, it's not increasing biodiversity at all. And using a virus to insert cystic fibrosis regulator gene into a person with cystic fibrosis. So this is a treatment for cystic fibrosis, but it's not going to be anything to do with increasing the biodiversity of cystic fibrosis. In fact, it's probably aimed at reducing the biodiversity by reducing the number of people that have cystic fibrosis. All right, question 14 and 15 relate to this information here, where basically we have students conducting a test where they're looking at whether chicken that's held in one condition has more microbes on it than chicken that's held in another condition. So question 14 was, which of the following is a variable that needs to be controlled in this investigation? So controlled variables are variables that if we don't control them, they will absolutely have an impact on our results. So something like the same type of nutrient agar used for each, that's something we want to keep the same because we want to see which of the conditions here, which of the variables of the temperature is likely to cause more variety of microbes to form. So if we have different nutrient agars for each different plate, then we've lost any control in that experiment. And so A is going to be the answer here. Let's look at the other options though. The same brand of incubator used. It doesn't really matter what incubator brand you use, as long as you're able to keep those temperatures at the same constant level. It could be a blue incubator, it could be a red incubator. They're not things that are likely to affect the outcome of the experiment. So that's not a variable that needs to be controlled. The same swab used for each Petri dish. Now, the idea here is we're taking swabs from different sources. That's actually a variable or part of our independent variable. So using the same swab is not a control that we want to have or a controlled variable we want to have for this particular experiment. And placing a nutrient agar plate without a chicken streak into the incubator. Look, we know that if there's no, we want to test the variety of microbes between the different pieces of chicken. If we put a nutrient agar plate in there that just has no swab on it at all, then probably nothing will grow. It's not going to tell us any information. So that would be more like a control rather than a controlled variable, but it's not really something that we need to have in this experiment anyway. All right, question 15. What is the dependent variable for this investigation? In other words, what is the thing that we're measuring in this test here? So the conditions the chicken piece is originally kept in is not what we're measuring. That's part of the independent variable. The number of microbes that were grown for each condition, that's certainly something that we could measure, but if we look at the question here, we're looking for the greater variety of microbes and not the greater number. So that means B can't be the answer that we're looking for. The size of chicken pieces used for each condition doesn't really matter. It doesn't really affect our experiment. Certainly not the dependent variable. We're not measuring the size of the chicken originally, but the types of microbes grown for each condition is what we're looking at. That's the variety of microbes. What different types of microbes are grown? Are there more microbes grown from one source than the other? So that's our dependent variable for this experiment. All right, question 16. In 2018, lung cancer was the most common cause of death in Australia. And in 2020, it's estimated there'll be 8,641 deaths. So this data is an example of deaths relates to mortality. So mortality is the answer. Incidence is new cases. Prevalence is total cases. Morbidity is how many people are suffering with the disease and have illness from the disease. But mortality is the statistic that relates to death. All right, question 17. The diagram below is a process during cell division. So the key part here is what's happening in this section here, which then causes this change in the chromatids as they get split into the gametes. So, type of cell division that's going on. There's only one type of cell division that relates in this chiasma and this recombination here. That's going to be meiosis. So our options are either A or C in terms of what's going on. Now you see this in sexual reproduction. It's part of the genetic variation that occurs through sexual reproduction. So A, sexual reproduction, C, asexual reproduction. We know it can't be C, so that means A is our only option. And just to kind of round out the answer, this picture here is indicating a recombination of the genes. So there's been some switching of the genes, and then the chromatid has been recombined. So it's got this new section of the gene with the old section of the gene. All right, question 18. The above diagram represents which of the following reproductive technologies? Now we have a plant, we have a paintbrush. We're taking the pollen from this flower and putting it on the stigma of this flower here. So this is very much typical of artificial pollination. It's not artificial insemination because there's no sperm and egg involved. It's not in vitro fertilization because, again, there's no sperm and egg involved. It's certainly not whole organism cloning because it is still a sexual reproduction process. So artificial pollination is the answer. All right. Question 19. This is looking at grey kangaroos found in a subalpine area. So this is all about how endotherms adapt to their environment. So if we're thinking subalpine, we're thinking cold. And so to keep warm in a cold environment, what type of fur are we likely going to need? We're going to need long fur. So A or C can be our options. Body shape and height, we generally want to have organisms that have a lower surface area to volume. That's how we deal with colder areas. So round and short is the option there. And then ear length, we want to have that short as well, reducing the amount of surface area that's exposed to the cold to keep the body temperature regulated. Ear length is short for both A and C, but when it comes to body shape and height, round and short is the answer we're looking for, and so A becomes our answer. All right, question 20. Fabry disease is a rare disease inherited in an X-linked manner. Xerophthalmia is a disease that's caused by vitamin A deficiency. And then mesothelioma is asbestos exposure. So you needed to classify each of these diseases into either environmental, nutritional, or genetic causes. So X-linked, we know from our hereditary unit that that's going to be genetic. So that means in terms of Fabry disease, the only option there that has genetic is option C. So we should know straight away that C is going to be our answer. But let's just confirm by looking at xerophthalmia, which is a vitamin A deficiency. Vitamin A relates to nutrition, not getting enough vitamin A for what they need. And then mesothelioma, breathing in asbestos, that's an environmental cause of a non-infectious disease. All right, so that's the feedback for the multiple choice questions. Hopefully that'll help clue you into not only how to answer these style of questions, but also to see where maybe you made some mistakes. And as I pointed out at the start, if we're able to improve our multiple choice results, then that can have a drastic effect in our final mark overall and could mean a big difference when it comes to your HSC result.